Good morning, everybody. It is 8:30 sharp, and we have a long day full of exciting events, so we're going to get started right away. I am Erik Lindahl, professor of biophysics and one of the members of the DDLS steering group, and I'm going to be one of your hosts today together with many other colleagues. But before we get started, I'm going to give the word to our actual host, who is Professor Olli Kallioniemi. He's professor of precision cancer medicine at the Karolinska Institute, heads Science for Life Laboratory, and in particular, the director of the DDLS initiative. Olli.
All right, thank you, Erik, and good morning, everybody. It's just a pleasure to see so many of you joining us today, and it's going to be an exciting day with the first ever data-driven life science conference. Just a few words on the program. So we are dividing the program into the 4 areas which represent the key application areas of the DDLS program, which I will now just in just a couple of slides illustrate to you. Many of you may know that already, but we have 11 partners, universities across Sweden who are having a life science involvement in them participating in the program. The program is funded by a generous donation from the Wallenberg Foundation, and it is run by the SciLife Lab board, or SciLife Lab board makes decisions. And we have close collaboration with the Wallenberg Centers for Molecular Medicine and the WASP program that I will shortly mention as well. So our vision is really that the future of life science is data-driven, and we need this type of a national program to accelerate the data-driven life science paradigm in Sweden and essentially change how life science is practiced in the future. We have many different elements and aims of the program that are highlighted in the strategy document that is available online. So I'm not going to go through that. Just show you a couple of vignettes of what has happened and what is ongoing. One of the major elements is the recruitment of young fellows, the young group leaders, the DDLS fellows, that there will be 39 altogether in the 12-year program. But now in the first phase, 20 PIs are being recruited, and this is ongoing. Roughly half of the PIs have been recruited by our partner universities, and I think this will be just a fantastic and wonderful group of young talent that will be joining the program. We've also run the very first round of joint calls between the WASP program, the Wallenberg Autonomous Systems and Software program, together with the life science program, and this resulted in 15 approved projects, but there will be many more calls of this type coming in the future. And just ongoing right now is the generation of the DDLS data platform, meaning a SciLife Lab data platform linked to the Berzelius supercomputing capabilities, but also data science nodes in these 4 areas that represent the areas that we will be discussing today as part of the program. So these will be sort of defined as a national role to contribute to the data science in these 4 fields. But this is just kind of the beginning, that overall in the 12-year time of the program, there will be many other aspects of recruitment, education, training, data-related, bioinformatics, data node-related activities. And many of them have not yet even started and will start many years into the future. So if you look at the year 2022, only some of the programs have been started and others will start later. And you have therefore time to participate, and we want you to participate. This is really a national program, and we look forward to collaborating with not just with the fellows and not just the PhDs and postdocs, but with all the departments and universities that can contribute to this program. So this is really an exciting joint effort that we hope to bring forward. And if I just sort of reflect this from the background of SciLife Lab, so you all know SciLife Lab as a national infrastructure, and we've had, therefore, a role in technology-driven research in the past. So now we can add on to it also a data-driven research angle in the future. Therefore, this is really a strong combination going forward and helping Sweden to stay hopefully at the cutting edge of both the technology and the data-driven life science domains. And with that, I will just wish you all very much welcome and sort of hope you enjoy the program and please interact with us and stay active. And with that, have an excellent program. I certainly look forward to it and all the presentations. And with that, I pass it back to Erik. Thank you, Erik.
Thank you so much, Olli. And I'm going to see if I can start to share my screen here. Just the PowerPoint window. The next step today, before we head on to our keynote speakers, is that we're going to have a brief introduction of the so-called research areas, because as Olli mentioned, DDLS is a gigantic initiative and it's going to become even larger. And to be able to organize this, we have 4 specific research areas, again, as Olli mentioned. The first one of these, I have the honor to coordinate, at least initially, and that is data-driven cell and molecular biology. So in cell and molecular biology, it's a traditional strength area of Sweden, and a whole lot of this is based not just on structural biology, but also computational biology going up to the tissue level. And here we really want to drive this more towards the data science point of view, by massive data generation, curating this data, enable us to extract knowledge from this data. And of course, as you will hear from all of these areas, this will involve a lot of machine learning, several unit recruitments working either on computational methods or in some cases focusing more just on building valuable data resources. One thing that personally makes me very happy about the DDLS initiative is that we've been able to invest a whole lot in young PIs focusing their careers more on method development or data development, because this has frequently been PIs that had it a little bit difficult to compete for funding when they compete with the experimentalists that only do the high impact papers. So as a couple of, I will not have time to go through all these things happening, but just as a few examples at SciLife Lab and the large infrastructures we have that generate data. Stockholm University started some massive investments in cryo-electron microscopy a few years ago that is generating tens of terabytes of data per day. But the challenge is, of course, how do we turn that data into these beautiful molecular images and movies and really biological understanding? That in this case, it's an example from Alexei Amund's group on the left. We have equally strong groups on light microscopy, and both of these things in microscopy, I think they're the second we put the sample under the microscope, after that it really becomes a data problem. And both of these are struggling with how to handle giga and tera and possibly petabytes of data in the future. And just a few weeks ago, we were able to recruit and actually get the first SciLife Lab fellow in this, DDLS fellow in this new research area confirmed. And that's going to be Joachim Bengtsson Palme, who's focusing on collecting massive amounts of data to understand antibiotic resistance and really sequencing things in lakes. You will hear a lot of our talks being about both analysis and prediction. You can hear Arne Ilofsson talk about some very large-scale stuff using AlphaFold to predict protein interactions. When it comes to cryo-EM, we have several groups working on really predicting motions by just extracting more data. And we have a number of purely theoretical groups, for instance, Jens Carlsson. Actually, Jens is not purely theoretical, but theoretical work using computers to, for instance, predict drug design. And I hope you're going to get a bit of potpourri from this from the talks, and then you will have a chance to talk more to us in the expert groups this afternoon. Janne?
Right, and I'm going to present the precision medicine and diagnostics area. My name is Janne Lehtiö, and I'm area lead for precision medicine and diagnostics. And for those of you that are not in the field, what precision medicine or personalized medicine, as it's called in EU, is about is to use molecular and imaging data to actually decide the best therapeutic options and preventive actions for each individual. So we actually characterize and aim to generate lots of molecular and imaging data to characterize the patients, to stratify this for the best possible treatments. And this is, of course, very data-driven process. So what we are talking about is to combine different data modalities like genomics and protein level data, imaging level data, metabolomics level data into a multimodal diagnostics, and then aid clinical decisions based on that rich data, and then communicate that to the healthcare professionals and also to the patients. So this is really a data exercise where you can connect lots of experimental data, but also data from the routine healthcare and the outcome data from the longitudinal and the population-based registries to learn about the molecular traits influencing the prognosis and so on. So this is joining lots of experimental efforts, but we are focusing on the data development here, coupling AI and different ways to push this area. So if you take the next slide. And as Olli said, we have been building lots of molecular profiling capabilities at the SciLife Lab at the technology platforms. And this new DDLS precision medicine and diagnostics area will allow us to put lots of emphasis on what do we do with that molecular data that we are generating on the cellular level, and then couple that to the healthcare professionals and clinical research that is done by universities and university hospitals to actually glue these things together, make use of the molecular data. So if you take the next slide. And of course, as I said, there is lots of areas of here. We have the molecular profiling data, we have the routine healthcare data, and enabling and catalyzing to access that. And there is, of course, lifestyle factors and environmental and socioeconomics that are influencing this. And this is, of course, huge area. There is lots of ethical issues. There is lots of legislation and policy issues that are hindering this type of research. So there is lots of Swedish challenges and opportunities in this. And this is something that we will discuss in the last breakout session in the precision medicine. So with that, I also welcome everybody to work with the DDLS Precision Medicine Initiative. And I think that this is one piece that can actually help the patients in future in Sweden and worldwide. Thank you.
Good morning, everybody. My name is Oliver Bilker. I'm a malaria researcher at Umeå University. I'm delighted that one priority area of the DDLS program revolves around the epidemiology and biology of infection. It is there's an obvious need there, of course, as we can all see in the current pandemic, to use large data sets here to help us battle significant global threats that come from these type of emerging diseases, but also, of course, from long neglected diseases and long appreciated problems like antimicrobial resistance, but also the now very apparent lack of antivirals and vaccines for many infectious diseases. We anticipate that as part of this program, we will build on the existing strengths in Sweden to computationally analyze and model host pathogen systems using multi-omics data sets and generate new hypotheses as to what might be good targets for vaccines and drugs. But then we will also use big experimental, clinical, or pathogen surveillance data and use them in innovative ways to understand how humans, animals, and plants interact with their pathogens and with their environment, and how these pathogens get transmitted through populations. And I just want to give a few examples of really big data projects that are ongoing. In Sweden and around the world, scientists are building global observatories to detect and monitor infections, both through immune responses of individuals, but also through pathogen sequencing. We're using systems immunology approaches by involving biobank samples, using genetic associations, or building on registry data at significant scale to understand and predict susceptibility and pathogenicity of infections. And we combine pathogen genomics and computational approaches also on the molecular side, like AlphaFold, to discover antibiotic and vaccine targets. And one example on the next slides is how, of course, Sweden got involved in making such data available through the COVID-19 portal. I think we will be doing more of that and help uncover data and make them available from clinical or biobank sources, experimental data, public health data, and then build on the existing strengths here in Sweden to provide advanced analysis capabilities and build an entire community that we can bring together and that consists of data producers and analysts. And I think this is a particularly fascinating challenge and opportunity that we have. Altogether, this will, of course, over the years improve the infrastructure and also the competence here to deal with current and future infection challenges. And on this last slides, I have just listed a number of questions, Erik, if you just move that forward. Exactly there, but I don't want to go through this. This is a slide I will bring up again at the breakout in the breakout room at the end of this conference, where I invite those of you who are interested in this subject area to join us and start to discuss and contribute perhaps to the solution of some of these questions and challenges that I've listed here. That includes also providing training and computational infrastructure and all that. So I welcome you all to not only to this conference, but also to a very active discussion around this subject. Thank you.
Okay. My name is Fredrik Ronquist. I'm professor at the Swedish Museum of Natural History, and I'm research area lead for evolution and biodiversity. So this is our definition of the research area. You can read it online if you're interested in the details. Intentionally, we've kept it broad in the hope of engaging the entire research community in Sweden in this research area. Next slide, please. Can I get, yes, please. This is a recent paper on avian diversity, which I think exemplifies beautifully how evolutionary biology is affected across the board by the data-driven revolution. So I took a few snippets from this paper. A recent wave of integrative studies combining comprehensive phylogenetic, environmental, and functional trait data at unprecedented scales is reshaping our understanding. And future expansion and integration of data sets may help to resolve some long-standing debates. Next. We see an exponential increase in data and data-based papers across the board. This is an example from the Global Biodiversity Information Facility, data in green and papers in blue. Next. This is an example of the scale of current projects. In the Life Plan project, biodiversity are collected at 200 sites across the globe for 6 years, using a bunch of different methods, yielding a total of 2 petabytes of data from this project alone. Next. Over the coming years, we'll undoubtedly see new techniques completely revolutionize our approach to grand challenges in our area. This is an example that's close to my heart. At current pace, it will take hundreds of years to complete the inventory of life on Earth. And we're now realizing that we can use machines to do this much faster, perhaps in less than a decade. This is using a combination of miniature robotics, automated imaging, high throughput sequencing, and artificial intelligence. And the figure illustrates what this might look like for insects. Next. Well, hopefully, DDLS will help drive these transformative changes in our area and join our breakout room at the end of the day, where the expert group in our area will present itself, and we will discuss challenges and opportunities in our field.
Thank you so much, Fredrik, and all of you. I will stop sharing there and let Ewan set up while I introduce him. So as all these speakers mentioned, you would have several opportunities to interact with all of them later. But the next step is I have the great pleasure of introducing our keynote speaker for the day, who is Ewan Birney. completely introducing Ewan would take half an hour, but the highlights here is that Ewan has been a pioneer in genomics and bioinformatics. He did his PhD with Richard Durbin around the turn of the century, and for the last 7 years, I think he has been heading EBI, which is the European Bioinformatics Institute, and since a couple of years, he is also Deputy Director General of all of the European Molecular Biology Laboratory, which I think tells something about how important bioinformatics and data is to EMBL nowadays. Ewan has been involved in numerous genome projects, and we might hear a little bit about some of them, but I just figured that in addition to being a Fellow of the Royal Society, he has received the Francis Crick Award, the Overton Prize, and the Benjamin Franklin Award, so it's a very impressive career. Ewan, we're very much looking forward to your talk, and it's great to have you here.
It's great to be here. I am only sad that I can't get to Stockholm. Someone needs to get me to Stockholm. I will get myself on a plane and appear in Stockholm randomly some point, I hope, this year, and see if I can meet up with many of you in person. So I can't get everything I want to get into this talk, but it's already wonderful to hear those presentations. I'd quite like to go to some of those breakout groups. I'm not sure I'll make them, but it just shows what a rich world SciLife Lab has. So let me just, can you see my slides? Yes, is that right? Is that good? Slides are there? Yeah, cool. So just to remind you about EMBL, EMBL, the European Molecular Biology Laboratory, we're an international treaty organization that does European scale science. We're headquartered in Heidelberg, Germany. Our second biggest site is EMBL-EBI, which is just south of Cambridge in the UK. And then we have two sites that are close to synchrotrons at Hamburg and Grenoble. We have a site in Rome specializing in epigenetics and neurobiology. And our latest site is in Barcelona on the beach, which is absolutely beautiful, doing organoids and tissue biology, sort of artificial tissues. Something like that. Okay. So we have 5 different missions across EMBL: to perform excellent research, to deliver scientific services, to do advanced training, that's PhD and postdocs mainly, innovation and translation, so we face industry as much as we face academia, and the integration of European life science research. And just this year, we have started our latest indicative scheme or scientific program from Edith Hurd, and this is called Molecules to Ecosystems. This is stretching traditional molecular biology, not just towards the organism, but going beyond that towards the ecosystems that these organisms inhabit. And that's both thinking about it from a planetary perspective, all the different bacteria and everything else in the wonderful world that of course have the same molecular rules of life as any human, for example. And also thinking about the human ecosystem, the ecosystem we create in our gut and the ecosystem we create with our behavior, which is human society. Now that's a bold vision. It is ambitious. We are not going to do all of this, but we want to be a leading part of the group of institutes that make that future happen. And just as you've heard about bringing zoology and other things much closer to molecular biology, I think there's just a huge opportunity here to make this real. In this new area of molecular biology, we kind of pull out, we still have our molecular core. So what EMBL is known for is fundamental understandings of molecules, sometimes to the atomic scale, up to cells and then organisms. But we have added on a number of specific areas called transversal themes. You can see that data sciences and theory are called out explicitly, as well as these areas of infection biology, human ecosystems, and others. And I just now want to go to the part of EMBL that I directly, which is EMBL-EBI. We are 100% dry. We only work in computers. We both, but we still fulfill all 5 missions. So we deliver data resources. That's the thing that we're most known for. When you use the human genome, you either use the genome from us directly, or you use the information that we provide by another party. So the human genome is just one genome, bread wheat, SARS-CoV-2, all of these different genomes come from us. And we don't only do genomes, we do what the genomes make, the RNAs, the proteins, how they fold up into protein structures, how they combine into pathways. And so that is a fundamental part of what we're doing. We do that with collaborators across Europe, including SciLife Lab in the context of Elixir, and we do it with collaborators across the world, including our colleagues in America and Japan. We also perform excellent research. We train the next generation of scientists, and we have a new program for training engineers in data science, not just scientists in data science. We engage with industry and help coordinate bioinformatics in Europe, and there our participation in Elixir and Eurobioimaging are key. So I just want to give you one sense of this transformation that happens, and I can't do justice to this, but anybody who's in structural biology, I'm sure, you know, that we've got a huge new computational tool on the table now that kind of goes along X-ray structure and EM, namely AlphaFold. Now, but if there's structural biologists in the room, don't, you know, I'm not going to, you know, this is a tool for structural biologists. It's not the answer. It doesn't solve, certainly doesn't solve all structural biology problems, but it completely lifts the game. And as many of you will know, AlphaFold, when it makes a prediction that it's confident in, it normally gets to sort of equivalent to crystal structure fit. And this is one of the fits in blue and green here between a crystal structure and the prediction. You can see some of the loop regions are out, but the absolute core of this protein is present. And John Malt, who runs CASP, says this is a quote here, this is the first time a serious scientific problem has been solved by AI. Now, what is interesting about that is, so John Jumper and Demis Hassabis and DeepMind were amazing, are amazing in creating this piece of AI, but they had to have 30 years of deposited data from the 1970s onwards to be in a position to make this computational discovery. And that goes to the interplay of how we what sort of data we collect and why we collect it, and then how we enable things to happen. And Demis and DeepMind came back to us at EMBL EBI and said, we want to make all of the AlphaFold predictions available to the community. Can you help us make that full set available? And that's why we launched AlphaFold DB with them, which I hope many of you use. Just to stress for the experimentalists here, the slightly freaky thing about computers is they scale stupidly well. And so this year we will do 1.9 million protein structures of all known UniProt sequences that we can get hold of. And so I think the benefit here is almost away from human, for example, where there's quite a lot of experimental effort, and on some of the other species or some of the kind of not, you know, backwaters, meandering parts of biology. Now, every molecular biologist can assume that there may well be a protein structure that they can use for design. So this, by the way, we've just released 200,000 new structures for the WHO's neglected tropical diseases. And as I say, we will come up to the full set by the end of this year across all UNIREF 90. Now, I just want to give that, I'm going to jump into the pandemic in the moment, but I also wanted to give a picture here about the great relationship EMBL has with SciLife Lab. And I think of SciLife Lab, and I use it as an example as a or the leading national coordinated lab in the life sciences that pulls together both data and experiments. I'm sure I at least got your up-to-date logo here on this. I was going to draw arrows between the left and the right, and then I didn't actually know which bit of SciLife Lab did which where, so I decided not to do the bijection between these things. But certainly there is this business of very clearly SciLife Lab serves Sweden extremely well. And it serves Sweden and it contributes to the European and international space. And at EMBL, we want to serve all of our member states across all of these different countries. And it's brilliant having a partner in SciLife Lab where we can interact with you. The COVID-19 data portal is a great example of that, where the Swedish one is in Swedish, which is great for public health. And it also can coordinate the deposition of data into the bigger databases that we run from that. And so just to remind you that EMBL is owned also by Sweden. So you imagine 6 little Swedish flags flying over these places in Europe that you can think of as part of the infrastructure that Swedish scientists can exploit. And what I would like to be in the situation is that any scientist from Lund to Umeå, and there's probably a town further north than Umeå, who wants to do something, they are just unconstrained scientifically. They can come to SciLife Lab, and you at SciLife Labs might say, well, I don't quite have that super resolution microscope, but I do know my colleagues in EMBL does, so let me help you go to EMBL. Or there may well be a data set they need, and the people in SciLife Lab can say, aha, I know exactly where it is. Here it is in EMBL EBI. So I really love this interaction between SciLife Lab and EMBL, and we, the two big frameworks in which those collaborations work are Elixir for the bioinformatics and Eurobioimaging for the imaging. But now I'm going to focus on a more kind of scientific area, which is how we respond to SARS-CoV-2. So the pandemic, we pivoted within a week. Actually, we started this in February of 2020, some of this, of pivoting some of our work towards the pandemic from research to services. And I won't go through all the different things. Oliver already mentioned the COVID-19 data portal, which is global, but we have national data portals, and you can see Sweden is listed in there. Our beamlines are still used as quality control for the BioNTech mRNA vaccine as well in the SACS beamline, for example. This is this portal which coordinates the fully open data set, not only around viral genomic sequences, but also in things like protein structure, small drug molecule interactions. And it's been critical to have these national nodes, which allow us to talk with public health officials and public health systems, country by country. And public health has a lot of priorities, and so making it easy for public health scientists to do the right thing about contributing to the global effort has been critical in this. So some lovely structural biology. We were one of the first places to do cryo-EM of the virus and the spike. As people know, it is rather flexible at the bottom. We've also looked at the replication, the polymerase of the virus and the main protease. But I want to now do a piece of research. This is from Maurits Gersten at EMBL EBI. He's now actually at DKFZ. And what I'm going to show you is a picture here of that Maurits generated actually in January 2021. February 2021. And what you're going to see on the right hand side is the distribution of all the other lineages of SARS-CoV-2. On the left hand side is B117. That is the kind of geeky name for the alpha variant, and you'll see that it will turn up at a particular time. So here we go, and you can see that there's going to be a hotspot here in Manchester, if you know your English geography. From other strains, and then notice down here there'll be a sudden moment where this pops up in Kent, and here we go, bang, and then it immediately takes over the UK. And having taken over the UK, that alpha variant then took over Europe and the world. And as you know, we're now on arguably our third or fourth, depending on which part of the world, wave of new variants that have come through the system. Now, underlying the ability to make such a model is quite a sophisticated piece of data science. So you have a data fusion problem where you're bringing together testing data, so the results of RT-PCR across the population, and sequencing data, which is a subset of this, and you have a model of how infection works to help model the noise in the data. And all of that means that you have to transform this data. You have two data sets, you have to transform them into a more appropriate space to do these fits, and then you can fit the different trajectories. And really, from that fit, you can understand both the evolution of the virus or the biological properties of the virus, which I'll come up to. But before you get to the biological properties, you have to understand the spatial correlation of how these viruses are working. And this is an example. Oops, I don't know why that's happened. Am I back? Is that good? Yes. You have to understand these spatial correlations because sort of random chance can occur in many places of just or variation in the way you control the virus happens for other reasons than the biology of the virus. But this is the key piece of inference that Moritz was able to do. This shows the relative growth rate where B1.1 lineage, which was an early lineage that left Wuhan, was considered to be the standard. So everything is done relative to that. You have all these different lineages, all these different types of branches across the tree. And then on the right-hand side, you can see B11.7. That is the alpha variant. And this was part of the evidence that said, or the strongest piece of evidence that said that we really had a different virus. It had a different set of biological properties. And therefore, the way we controlled the virus previously was not going to work in early 2021. And that information we immediately released to many different countries around the world. And we were particularly involved in the discussions in the UK. My colleague Rolf Ampfweiler presented to the Bundeskanzleramt, the Chancellery, in mid-January, and we presented it to the scientific leadership in France as well. We did, just to reassure you, we did email Sweden and Tegnell and everybody else as well. So it was an absolutely generous offer of our insight into what was going on. And I won't, this is some of the backup here about the fact that the biology really had changed. It's actually quite hard to remember when you go back to that time, because it was the first time, people were quite skeptical about the biology of the virus had changed. And they thought that it was just bad management by the UK. Now, the UK had some very bad management moments. So that's not to say that the UK didn't have bad management moments, but the biology of the virus really had changed. And here's some of the supporting evidence behind that. Now, I was pretty amazed that we did all of that. Moritz came up with that in under 2 weeks. It's kind of amazing, that sort of speed of turnaround. And then we had to do it all over again for Delta. And for the aficionados in infectious biology modeling, there's this problem. Delta originated in India. And we had transmission of Delta strains from India into Europe, but in particular in the UK. And we had to balance whether the increase we were seeing in Delta at the start, was that due to higher migration, or was that due to a different piece of biology? And this, again, being able to infer robustly from real world data what is going on is harder than it looks. It's not as simple as just plotting how much delta you have. And on the right-hand side here, you have a picture of delta in this brown line of its daily cases. And the important thing to notice here is the model is allowed to start not at 0 or not down almost at 1 on the right-hand side. And that is an important piece of sort of data science innovation about how you model migration versus growth in these models. Now, all of this, I've got other examples here of how we have worked in with the SARS-CoV-2 pandemic, but I want to generalize this. And I want to generalize this into one of the big opportunities for our field, and that is bringing together the world of healthcare operations and research closer. So these two areas are very distinct. They do very distinct things. Healthcare operations is about delivering healthcare. Research is about understanding what is going on in the world. But there is a huge amount of synergy, and to release the synergy, you have to think quite carefully about it. And just if you look at this in the COVID-19 world, so for healthcare, you have operational healthcare data. Everything has to happen fast, really, really fast. There has to be robust analysis that will run every single day or every hour sometimes. And yet you need to be able to integrate new ways of thinking about, in this case, the virus quickly. On the flip side, the research delivery is a sort of medium to slow turnaround versus the sort of daily turnaround of the healthcare operations. There's this thing that slightly freaks everybody out about researchers, which is we sort of don't know precisely what we're looking for when we go into data sets. And that freaks out our healthcare colleagues, and we have to do a lot of reassurance that we're not just like going to fool around. We know what we're trying to, we know what the end goal is. One thing about research is that we are able to, usually you have to understand everything that's going on in the system to be able to extract the biology, and that leads to new QC measures. And one of the biggest benefits of bringing research alongside healthcare data is you get better QC methods that come from research over to healthcare. And of course, then you create new analysis schemes. Now, I just gave that example about the SARS-CoV-2 example, but actually a longer term example is genomic medicine. And again, you can write down exactly the same sort of problems on each side of this world in the same way, where genomic medicine is has operational healthcare needs, and it's going to provide diagnosis for rare disease. You've got more time there, but also cancer. Now there you've got probably days or maybe a week or so. So everything has to go very, very quickly. At the same time, you've got this research aspect with exactly the same aspects of doing research, but I would like to stress again, when you bring researchers closer to this operational data, you actually get better and deeper QC on your operational healthcare data. And this is one of the reasons, as part of the new program, we have what's called the Genomic Medicine Platform. This has 5 components: providing strategic advice. This is basically me and a couple of colleagues who can talk to people who want to set these things up. We have quite a bit of experience now. As I mentioned, we provide the reference human molecular data set of the human genome, its protein structures by AlphaFold DB and other things like that. There is the EGA Federation, and I know SciLife Lab runs a federated EGA node, or is thinking about a federated EGA node for handling this. We can run technical training courses, on site, certainly by Zoom. And we are looking to add mid and long-term secondments as well, where engineers can come and spend time at EBI on a jointly agreed project, and therefore upskill their data science, bioinformatics skills to go back. So that was a whistle-stop tour. I hope I've got time for questions, and just to say that we would hope to work together for Europe and the world, and let me repeat that I'm just sad that I'm not in Stockholm. So thank you very much for listening.
Thank you so much, Ewan. With 500 participants, I don't think we have a chance to take questions for everyone, but I can ask you one thing, that Sweden too is of course a society that we learned many things through the pandemic, and I don't think anybody did everything right here. And you alluded to this a little bit in your slides, that I think naturally, both in medicine and healthcare, the natural reaction, right, is that we don't want to apply things in care until we know for sure. And that's very human, and I think most MDs would admit. And on the other hand, there is this challenge that in particular for a virus that's mutating rapidly and everything, the earlier you act on data, the better it will likely be. And I guess this is a universal challenge. How are we able to feed this very uncertain data that researchers have probably learned to live with, that we can handle with statistics or something, but getting primary care physicians or doctors to accept that they're going to need to base treatment on things that they actually do not know to 100% certainty?
Yeah. I mean, I think that's true, though I actually think for me, the major thing that I learned was the success of the recovery trial in the UK, where one of these, these kind of new ways of doing randomized controlled trials, where you just set up a trial that goes effectively forever. Okay, so your breast cancer, you say, we're just going to just recruit people forever, and then you add and subtract trial arms as you see whether things work or not. So I think the key learning there about healthcare, about rapid healthcare innovation, is that you should set up one of these modern, I mean, they're actually very simple, and they're not sophisticated, they scale, and you just literally loop through every single thing that you want to try very, very quickly. Because if it's urgent, you will have enough cases coming your way, and then you will be able to make a decision about this. So I think for a lot of healthcare, whenever you can set up one of these robust trials, that is a good thing. There is another aspect which I think, I think every country struggled with, to be honest, having interacted with all countries, which was the interaction of data and modeling and policy choices. And that interaction was very complicated. Many people would say that we needed to integrate our understanding of how the pandemics work with an understanding of how economies work, because some of this had an impact on the economy. And I think many people could claim that the Swedish set of decisions had a better inherent balance between the economy side, or the well-being side, and this, economy in its broader sense, about being nice to that, having a good life, as it were. But actually, when you actually delve down in that, before you get to the economy side, most of these models didn't even work between operational healthcare and infectious epidemiology. Like, how do I have enough hospital beds coming up now, or in 2 weeks, or in 5 weeks, or whenever? And we have people do model that side, and they model the infectious side. Actually having a joint model that does them together is rare. So I think there is a piece of establishing research that stretches from kind of data science, infectious molecular biology, infection biology, and data science areas of molecular biology through operational healthcare. It's kind of effectively health economics and epidemiology, somewhere between those two disciplines, and then also getting to econometrics, or not economic, economic policy, social studies in some sense. And I think when you look at that, we just didn't have people who could talk the language across those three fields. And that was a failing collectively, that we hadn't made those links beforehand. And the problem is that when you're in a crisis mode, what you can't say to someone is, look, could you please study this field, these two fields, for 2 years and be able to transition between the two of them? Everybody has to learn on the job. So I think there was a gap there. It's not to say there aren't epidemiologists who know a lot of molecular biology, there are, and vice versa, but we didn't have joint models. That would be my critique of this. We need a joint modeling institute ahead of a crisis.
So not just driving the research by data, but driving the entire decision process very rapidly by data.
Yeah, and again, it's not the decision process. You still have to have wise people who present things to politicians, and politicians still have to make decisions. The decisions definitely have to be made. The model will not spit out decisions. And actually, talking to some of my colleagues from econometrics, like the real people are saying, the Bank of England, who set the inflation rates, they have models, the experts don't necessarily always trust the models. Yeah, but what the models do is sort of allow you to explore the data sets and explore the future. And then sometimes experts say, this model is just plain wrong. I'm really sorry, but we're not gonna base ourselves on that. But at least you should do it with that ability to model and explore. Like the human head cannot hold in its head how, you know, two exponentials will, with some interaction term, will really work. So I don't want to, you know, it doesn't become so technocratic that there is one model that you just run. But what I do think we were missing were experts who could live in the infectious epidemiology world, could live in the health economics world, and could live in the kind of macroeconomics world of policy. We just had nobody or no teams that could move between those three areas.
Thank you so much once again, Ewan. I think we're going to need, let's all of you join me for thanking Ewan, at least with the virtual hands. Thank you so much. And that brings us to our first break / leg stretcher / bio break. We're going to reconvene at 9:25. We're going to have 3 talks within the area of cell and molecular biology. So take your 5 minutes and grab a new coffee and then rejoin us. Thank you. All right, welcome back everybody from a short break. The first research area specific session is going to be on cell and molecular biology, and you're not going to hear so much from me because we are going to focus on our 3 great speakers we have today. The first speaker I have the pleasure of introducing is Orane Guillaume-Gentil from ETH Zurich, who has worked with Julia Vorholt for a couple of years, and in particular integrating data over several length scales and being able to measure things inside live cells, which is pure science fiction to me, and I'm in shock and awe that this is even possible. Orane, it's great to have you here, and we are very much looking forward to your talk.
Thank you very much. And first of all, good morning, everyone. And thank you very much for this nice opportunity to share our work with you today. And yes, so in this presentation, I would like to present a method we recently developed, which we named Live-Seq, which basically allows to profile the transcriptome of single cells, but without killing those cells. And so, as you know, current approaches in single-cell transcriptomics generally require to first isolate the single cells and then to lyse these cells in order to access to the endogenous molecule or the transcript here. And so because of this, all these current approaches are endpoint measurement, providing snapshot data. Now, of course, there is an interest in biology to get some temporal information, and it would be great at this single-cell transcriptomic level. And because of this, there have been in the recent years many different strategies and protocols that have been developed, including computational approaches to infer some cellular trajectories, or also strategies that label a cell and its progeny, or label some of its molecule. And all these methods allow somewhat to use snapshot data measurement and infer or model some temporal information from those one-time point measurement. But now with this Live-Seq approach that we have developed, we can use a microfluidic probe to sample the RNA from the cell. And there we sample only a fraction of the cytoplasm of a cell, which we can then redispense out of the cell for further processing and the analysis. And with this, the particularity of this approach is that the cell is not destroyed in the process, but it remains alive. And with this, we can now access the downstream molecular or phenotypic state of this same cell from which we have acquired a molecular profile. And so instead of inferring some molecular history based on one time point measurement, we can now directly measure the same cell at different time points. And so to do this, we are using this FluidFM technology or fluidic force microscopy technology, which is basically a miniaturized pipette that is matching the scale of individual cells. And so you see here in terms of the volume that we can handle with this technology, we are in the picoliter and femtoliter range, so 1,000,000 times smaller than what can be usually handled, for example, with the glass micropipettes. Now, beside the volume, the probe itself is also small, so we have a length of about 200 micron, and the probe is featuring this pyramidal tip, which is about 5 micron. The whole probe is then hollow, and on this pyramidal tip, we have a small aperture, which allows to exchange liquid between the inside and the outside of the probe. Now you see here the whole setup. We have our probe. In the back of this microchannel, we have a digital pressure controller to regulate the pressure to flow liquid into the probe. And to operate this probe, we use an atomic force microscope. So in short, this make this pipette force sensitive in a system where this force sensing is coupled to a piezo-driven displacement of the probe. So we can very precisely and sensitively operate the probe with this atomic force microscope. Now, in addition, the whole setup is mounted on an optical microscope, so we can also monitor in real time all this manipulation that we are doing. Now, to extract such a cytoplasmic biopsy from a living cell, we developed the following method, where we first align the probe on top of the selected cell and the position on these cells where we want to insert the probe. And then we can use this force control to drive the probe into the cell and then maintain it at a constant force inside the cell cytoplasm, so that we can, during this time, apply under pressure to flow cellular fluid into our probe. And when this is done, then withdraw the probe out of the cell. Now we do those manipulation with probes that were pre-filled with mineral oil, and this allows them to confine this cytoplasmic fluid in the front of the probe, and also to visualize in real time this cellular fluid. So we see here an example during the extraction of this cell. We are inserted in the cytoplasm here, and you see this front of the probe is now filled with some cellular fluid, while the back is filled with mineral oil. And so we can not only visualize this in real time, but we can also quantify it because we know the dimension of our probe. And so you can also in real time know how much cellular fluid you are collecting in your probe. Now, this is another example, this time in fluorescence microscopy, where we monitor the extraction of these lower cells. Those are 2 cells expressing soluble GFP. And sorry. No, sorry, I cannot run the movie, but what we would have seen is how we can deplete this soluble GFP from this cell and collect it in our microfluidic probe. Now, once we collected this cytoplasmic fluid into our probe, we can also redispense and to transfer this picoliter extract onto a variety of analytical substrate. So we won't go into the details here, but we could show that we can address a substrate requirement for different analytical methods downstream, and we were able to perform a few proof of principle molecular analysis on such cellular biopsies, which show that we can sample and then also detect and analyze the endogenous proteins, metabolite, or transcript using this approach. Now, those were preliminary, so to say, molecular analysis. And in the recent years, we wanted to push those bioanalytics a little bit further. And so we have been focusing on transcript and trying to move from these few qPCR assays to a genome-wide RNA sequencing based on these cellular biopsies. Another challenge for not only for RNA sequencing, but any analysis of such cellular biopsies is, of course, that we have a very low amount of material to provide to the analysis. So only a fraction of a single cell. And we see here for RNA sequencing, we started with the SmartSeq2 approach, which was the most sensitive single-cell RNA sequencing approach available. And what we see here on this plot is, depending on the input of RNA we give and process with this protocol, how much cDNA we can yield. And we see that the approach is very efficient for a single cell amount, so between 5 to 10 picogram of total RNA. But if you have less than this amount, like we expect for our cellular biopsies, then this protocol is completely failing. And so our collaborator have been working on optimizing this protocol, so that now you can see here in blue, we have a protocol for RNA sequencing that allows to increase this sensitivity, and so that we can now analyze input that are about 10 times lower than the content of a single cell. And having this protocol, we can then analyze cytoplasmic biopsies. And so in these studies, we've been working with 3 different cell types here, and 2 of the cell types, we also had 2 different cell states. So we have here in brown primary stem cells that we analyzed before and during differentiation. We have a fibroblast cell line here in blue, and in green we have a macrophage cell line, which we also analyze unperturbated or stimulated with lipopolysaccharides. And we see that with our protocol, we can indeed nicely obtain genome-wide transcriptome from such cellular biopsies. And here in that case, yield 5 different clusters that were mainly driven by those different cell states and cell types. And we of course perform a lot of other quality control on those data and the usual differential gene expression analysis, blasting the marker genes to the mouse gene atlas, etc. And all this data confirm that the transcriptome that we are measuring with this approach can accurately stratify the different cell type and states that we have been testing. Now, after this, we also compare those Live-Seq data that we measure to the standard single-cell RNA-seq data. So here we measure with SmartSeq2 entire cells of the same types and states as we have in our Live-Seq data set. And so then we could integrate both data set shown here, which revealed that we then again find our 5 different clusters, mainly driven by the different cell type and states, but no obvious molecular difference between the different approaches that we use. So we have here in red our Live-Seq data, in blue the SmartSeq2 data, and we see that they nicely integrate and that we are not having measuring bias data with the Live-Seq approach. Now, for this approach to be of interest in the field, it has to first keep the viability of the cell, which we have now assessed for different cell types shown here. We have the 3 cell types used in this study, and also HeLa cells that we were working with earlier. And what we saw is that for all of these cell types, we have the vast majority of the cell that survived the extraction, so between 85 and 89% here. So the majority of the cell remain alive after the extraction, and this is also true not only for cell line, but also for primary stem cells, also for these macrophages here that are relatively small cells. Now, keeping the cell alive is not really enough to be able to monitor a biological process downstream, so we also need the cell really not to be perturbated, or at least to be able to characterize the perturbation. So we have performed a few other assays to evaluate what is happening to the cell after the extraction. And here first is a few observations on cellular function after the extraction. So working here with those macrophages and how they respond to stimulation by LPS. We use a reporter cell line from this publication, which has a fluorescent reporter for one of the cytokine that is expressed in response to the LPS stimulation. And with this, we can quantify and monitor a single cell for the expression of TNF-alpha and plot some profile, so TNF-alpha response profile for individual cells. And so we've done that for control cells that were non-treated or treated with LPS, but also, of course, for cells that we extracted, whereby we extracted the cell either before stimulation or 4 to 5 hours after the stimulation. And in that case of this example profile shown here, we also extracted some cells both before and after the stimulus. And what you see in this example is how the cell is extracted twice, but still is really responding to the LPS stimulation and producing this cytokine. And so we've done this for more than one cell, and we could show that altogether the cells that we are extracting were all still responding to the LPS stimulation in a way that was similar to the cells that hadn't been extracted. So altogether, we couldn't detect here any major perturbation of this particular cellular function. Now with these macrophages, we were also able to profile the volume growth of individual cell over time. And this is because those cells are semi-adherent, so they have this spherical morphology. And with this, we can, without any perturbation, we can measure cellular volume over time. And this provides us with this kind of profile. So we see here a control cell that was exposed to LPS, but not extracted. And you see a relatively noisy measurement, but it is sufficient to monitor how the cell volume is increasing along the cell cycle. And now, more interestingly, we did that for cells that underwent the extraction, as shown as this one in this one example here, where we can monitor the growth of the cell volume, and then we see the drop in volume when we extract the cell, where we can relate also the volume that we measure in our probe that is matching this volume that is lost by the cell. And then interestingly, we can then monitor how the cell react after the extraction. And we see here a relatively steep growth of the volume, with the cell recovering its initial volume within a relatively short time. And after this, the cell volume growth continues in a similar pattern as what we observe from non-extracted cells, with in some cases also cell division, like shown here, with a drop of 50% of the cell volume, and then 2 daughter cells with overlapping profile here. And so with this few cell volume profile measured, we could show that this, so here from the smallest cell types that we have been working with, we extracted between 40 up to 70% of the total cell volume, and those cells were still alive and growing apparently normally. But those cells needed first between 100 up to 300 minutes to recover this volume the cell had before the extraction. Now, finally, we also had a look at the transcriptome of cells that underwent extraction. Here we worked with IBS cells, and we extracted cells and then waited either 1 or 4 hours to process the cells with conventional single-cell RNA-seq, so that we can then compare the transcriptome of non-extracted cells here in red together with cells that had been extracted 1 or 4 hours before. So this 1 and 4 hours are time point where we were expecting to see some transcriptional response to the biopsy. And so first, in this 2-dimensional projection, we couldn't see any difference between the 3 different conditions. Then even in the differential gene expression analysis here, we could detect only 12 genes that were mildly under or over expressed in comparison to cells that did not undergo extraction. So no major transcriptional response. However, what we saw in these 12 genes is that about half of them were somewhat related to the cell cycle. So we also looked at so making the cell cycle scoring from those measured transcriptome. And we didn't synchronize the cell or anything, so this is to take with caution. But what it seems to show is that here we have the population of cells that didn't go undergoing to extraction. And compared to those that were extracted, it seems that we are losing some cells in the G2M phase and accumulating more cells in the S phase. And this is in relatively good agreement with this time needed for the cell to recover this volume here. And so if you would take all these cells and drive them, each of them 2 to 3 hours back in the cell cycle, you may indeed lose cells from the G2M phase and accumulate more cells in the S phase. So altogether, no major transcriptional response to this biopsy sampling, but the only thing we could detect so far is a minor, potentially a minor cell cycle delay following extraction. And so having no major perturbation, this now opens the door to dynamic measurement. And so the first thing we did is to see whether it would be feasible to really sample the same cell twice and measure its transcriptome at 2 different time points. And so we started with this model system of the macrophage response to LPS stimulation, which is a relatively fast cell state transition from the non-stimulated to the activated states. And what we did is to sample a cell a first time before the LPS exposure. Then we added the LPS and waited 4 hours to sample again the same cell, this time in the responsive state. And so we managed to obtain 4 paired transcriptomes, so 4 cells for which we had both time points. And then we could project those data on our larger data set with all the Live-Seq and single-cell RNA-seq data. And we could control that all the measured transcriptome at the first time point were clustering correctly with the non-stimulated macrophage. At the second time point, then clustering together with the LPS stimulated macrophages. So those gene expression profile with this sequential sampling correctly mapped to their respective cell state cluster and showed directly here on this 2-dimensional projection, this transition from the basal to the LPS stimulated state. Now we did then the same with another model system, here the adipogenic differentiation of our primary stem cells. So first showing that this is also doable not only with cell line, but of course also with primary stem cells, but mostly showing that we can work at this few hours time scale, but also at a longer time scale here, several days apart. So we sampled the cell the first time to monitor the transcriptome before differentiation. Then we added a chemical cocktail to induce the differentiation and sampled the same cell again, this time 2 days after. And with this, we obtained 8 paired transcriptome, which we could again project on our larger data set. And again, we had the first time point correctly clustering in the non-differentiated cell cluster, and the second time points in the differentiating ASPC cluster. So same as before, we can now also directly map this cell state transition on our t-SNP plot, showing the transition from pre to differentiating states. But in addition to the previous experiment, in that case, it's a little bit more challenging to monitor cells over several days. And so we also added a genetic barcode to those cells so that we could also recover those barcodes and show that we are indeed really sampling the same cell at the 2 different days. Now, besides sequentially sampling the same cell at 2 different time points, we can also use our Live-Seq approach as a transcriptomic recorder. And the interest here is that we know that most cellular response are heterogeneous, with cells responding weakly or strongly to the same stimulation. And this is true for this LPS response of the macrophages, where we see here with our fluorescent reporter for TNF-alpha, the large heterogeneity that can be characterized in the responsive states. But now what the Live-Seq approach allows us to do is to record the transcriptome of individual cell, this time at the basal states. And then we can apply the stimulation and this time look at the cellular response to this external stimulus here using this quantitative time-lapse imaging. And so with this, we obtain a dual data set where we have the ground state gene expression of an individual cell linked with its downstream cellular response, and we can look whether there are molecular factors that can underlie this heterogeneity that we can see during the response. And so we obtain here 17 dual data sets with this experiment. And what we could do first is an unsupervised genome-wide ranking of individual gene expression at the ground states of this macrophage as a function of how much they impacted this downstream LPS response. And what came up out of this genome-wide ranking is this NF-kappa B alpha inhibitor, which was the major phenotypic determinant, and showed a negative correlation. So the higher is the level of this particular inhibitor, in the ground states, the less the cell will respond to the LPS downstream. And so another thing that came out of these mini studies is again with this cell cycle scoring from those measured transcriptome in the ground state that shows that the cells that were in the S phase of the cell cycle at the time of the stimulation did also respond weaker to the LPS response. So both of these results could be then validated with independent experiment, and so our Live-Seq data was able then to identify the basal NF-kappa-B alpha inhibitor expression level, as well as the cell cycle state that were important phenotypic determinant of this heterogeneous LPS response. And so with this, to summarize, we have developed this Live-Seq approach that allows to sample cytoplasmic biopsies from living cells, from which we can then measure genome-wide transcriptome profile, which accurately can stratify diverse cell types and states. And they can do this without inducing major cellular perturbation, which then allows to measure or start measuring now cellular dynamics, as we showed with the sequential sampling of the same cells at 2 different time points that allow a direct mapping of true cellular trajectories, but also as a transcriptomic recorder, whereby we can investigate the impact of ground state gene expression on the subsequent cellular response. So with this, I would like to thank the group of Julia Vorholt, where I am working currently, but also the group of Bart Deplonk, with whom we collaborated to develop this Live-Seq approach. And of course, thank you very much for your attention.
Thank you so much, Orane, for a truly exciting talk and a fascinating technique as a look into the future. We have a couple of questions already in the chat here that I will read in the interest of time. So Teresa Frisan is asking whether you see any upregulation of mRNA for P21 or P16 in association with the cell cycle arrest after sampling.
So we did not. No, we did not. But yeah, doesn't mean that we. Yeah, so I'm not sure how much we can detect if the effect is not major, let's say, from single gene.
So what kind of resolution would you expect to see? That would it? Are you talking that you would need 50% or so to detect anything? Or?
It depends. So typically, this cell cycle scoring also is then the group of gene where we are able to detect a little bit more. I think altogether we detect in average 4,000 genes with Live-Seq sample versus 8,000 for a single cell. So one thing is that in this study we couldn't focus yet on one particular biological process. So here we have 400 transcriptome data, but split into the different state and types and conditions. And of course we can help by increasing the number.
I might come back to that shortly with a question of my own. But first, Jianqing Hu had a question whether you have compared differences of the biopsy extracted from different parts of the cell.
Not yet. So what we see optically with some labels is for small molecules like the transcript, if they are soluble, we can collect them from the cell periphery and the whole cell, basically. We never tried this on neuron or larger structure where we could really analyze this spatial location of the transcript. But for a standard average adherent cell, for instance, we see the extraction of throughout the entire cells and not really restriction. In another work where we go to extract organelles that are much larger, there we see a local effect and the extraction of the organelles only around the small area around the tip. So there's probably ways to tune this and be able to extract only locally, but we have not been working on that. And with the current approach, we extract from the whole cell.
Joel Hedlund here has a question about the micromanufacturing of the Afm Pipettes and whether you did that in-house and what the most challenging parts were there.
Yeah, it is indeed difficult to produce those probes. And now the fluid FM technology is commercialized, has been for at least 10 years now. And we have here these 3 companies, so Cytosearch commercializing the instrument. And this company, SmartTip, now belonging to Bilker, is producing those probe. Typically, to make this single aperture nanometer size on the pyramidal tip, this is now doable with the microfabrication, the wafer scale, but has been a limiting factor for a long time. So sometimes we do ourselves process those tips. So in that case, we mill the aperture ourselves, also while developing the protocol so that we can tune a little bit the properties of the probe. And once we have developed protocol, then the companies come behind and develop standard probes to make them commercially available. But it is, challenging.
Cool, but that, in theory, that means that one could buy it from these companies. That sounds good. Yeah. Ola Larsson had a question whether you can extract RNA associated with ER.
So from the, I don't think that we are shearing and tearing apart larger structure. But what we can now do is to use another probe design with a larger probe, which we use to manipulate organelles. And we have shown that it is indeed possible to extract part of the ER as well as part of the mitochondrial network, which we can now extract and then even transplant into other cell. So we haven't performed molecular analysis with those new approaches, but in theory or conceptually, it would be possible, yes.
And I think we have one last question here from Yawen Wu, and he wonders what the coverage of the whole transcriptome is in those 40 to 70% extracted cell volume.
So I don't have a number here, but we did compare the overlap with single-cell RNA-seq, which was around 80% or 86% or so. And we had some some genes that we detected only with single-cell RNA-seq or only with the Live-Seq. And we could show there that if we downsample the single-cell RNA-seq, then this overlap is increasing a little bit. But altogether, it's so we could show also that there is no bias, so there was not really a subset and an enriched geoterm that we measured only with the Live-Seq approach, or that we missed with our Live-Seq approach. So in general, a relatively good overlap with the single-cell transcriptome measured by other methods.
Thank you so much, and I would encourage the audience to join me with a virtual applause, at least for Orane 2. Thank you so much for a truly exciting talk.
Oh, thank you.
Our next speaker is Aleksandra Walczak, who has a background more on the theory side and in particular exploring networks. And she's a professor at the École Normale Supérieure, and we are very much looking forward to hear your talk too, Aleksandra. It's great to have you here.
Thank you. Let's see if I can now share screen. Yep. Okay. Here we go. So thank you very much for the invitation and the opportunity to speak. So I will talk about work about the immune system, which we've been doing in collaboration with Thierry Marat, also here at DNS, and many collaborators, both theoretical and experimental, from all over the world. So let me start with a whodunit. And so imagine a scientific meeting pre-COVID in a remote location with excited scientists and an amazing after party. And the next day, Joe is found dead. And next to Joe's body, there are a few drops of blood that do not belong to them. And this being scientists, they cry genetic tests for everyone, and genetic tests reveal that the murderer is one of two twins, either Alex or Terry. So traditional genetic methods, sequencing methods, cannot distinguish between identical twins. So they call in Detective Asia Bear, who has access to more modern techniques based on the immune system. So we all have, as you're very much aware after these last couple of years, an adaptive immune system, which is made out of B and T cells. So these, for example, T cells, but B cells essentially work the same way. They have receptors on the surface, and these receptors have the ability to discriminate, bind molecules, and based on that discriminate whether this molecule is natural to your body or whether it's foreign to your body, in which case you should initiate the immune response and get rid of it. So recently, thanks to sequencing technologies, you can just get a blood test and we can sequence out your T cells and, we can sort out your T cells and sequence them. So basically get the reads of the proteins that code up for these receptors. So
Aleksandra,
since every
Can I just interrupt quickly? Could you remove that build order window? Because I think people can't see the lower part of your screen on the right.
All right, let me, I actually don't see it. That's the problem.
Okay. I think if you just close the build. Yeah. Yes, is now.
That fine?
Perfect. Thank you so much.
Okay. Yeah, sorry, I couldn't, I don't see it. So it's, right. Okay. So we have an immune system. The way it works is that if we encounter a pathogen, this triggers an immune response. And basically what that means is that the cells that recognize the pathogen proliferate and make many copies of themselves. So then if we actually have insight into the composition of the immune system through, say, sequencing, we can figure out what is in principle, we can figure out what is the set of infections that we've encountered. The only problem is we actually need to learn how to read this amazing personalized medical record we're all walking around with. And so that's the difficulty, and that's what we're trying to do. So, okay, so how can this help us solve the murder mystery? So by comparing the drop of blood found by Joe to drops of blood coming from different people, we're actually able to figure out who this drop of blood belongs to. So in which case the two samples belong to the same person, and in which case they belong to two different people, just by looking at the number of shared sequences between people. Because if the drops of blood, if the samples come from two different people, from the same person, they'll share many more unique T cell sequences than if they come from two different people, even if these two different people are identical twins. So here we see identical twins in pink and any other set of two different people in orange, and you see that there's very little difference, whereas there's significantly more sequences between twins. This is actually stable over time. Even here it's shown on the times of days because that's what we have data for, but we can computationally show it's stable over years, and this identifies the murderer as Alex. But the question is, how does this work? So how many sequences, T cell sequences, do we expect to share? So in order to see that, we have to go into the guts of this process. So T cell generated. So the first thing to realize is you have 10 of the order of 10 to the 9 different T cells. If you wanted to encode each one of these receptors as a separate protein, so that means encode via a separate gene, this would not fit into your genome. You have otherwise of the order of 10 to the 4, 10 to the 5 genes. So this is a few orders of magnitude more. So nature has come up with a very funky way of generating diversity in T cells and B cells, which is to combine combinatorics and randomness. And so the way this is set up is that, to simplify, each T cell is made out of 3 different parts, molecular parts, which are called V, D, and J. And in your DNA, in each of your cells, you have a number of templates for V, D, and J, a number of genomic templates. And then randomly you pick one V, one D, and one J, and you actually edit the DNA in these lymphocytes, in these immune cells. And then additionally, you insert and delete nucleotides at the junctions between the V and the D and the D and the J. So the combinatorics, if you look at these numbers, gets you to the order of 1,000 different combinations. Adding these insertions and deletions gets us to the order of 10 to the 9th. So you can imagine basically you're putting words together and taking letters in and out. If you do that, you can end up with something that's complete rubbish. That happens, but you have 2 chromosomes. So you have 2 shots at this working in each cell. And it turns out that if you look at the sequencing data, some fraction of these reads are out of frame, meaning they come from a failed recombination event. And thanks to the fact that they're out of frame, that means we know they can never be expressed and work as functional proteins. So all the selection, all the proliferation that happens later that I described cannot happen to them. Thanks to that, we have insight into the generation process. So we know that the reads that are out of frame belong to sequences that only underwent generation. And so our name of the game now is to figure out what's the probability of generating a given T-cell sequence. What's the probability with which your body generates one? And so what we did is we did a probabilistic algorithm, figured out a probabilistic algorithm to calculate the probability of generating a given sequence. In short, for each sequence, we consider a library of all possible scenarios, by which I mean V choice, D choice, and J choice, and the number of insertions and deletions at each of the junctions. We consider all the scenarios that are compatible with the observed sequence. Then we assign weights to them, and for putting together all the sequences and all the scenarios, we calculate marginal distributions, such as the probability that a given organism uses a given VG, has a certain number of deletions, insertions, and so on. And then we have a model, which is the most general factorializable model that we use to recalculate these weights. And this is an expectation maximization algorithm, which is bound to converge, and it converges, it gives us back stable weights for each of the scenarios. And by summing up over all the scenarios, we can calculate the probability of each sequence. So in short, either you know what you understand that, or we have a way for calculating the probability of that your body generates each of these sequences. So what that means is we can then study these probabilities. And the first thing to notice is we can make a histogram of the density or the probability of how many T cells do you have that have a probability that's 10 to the minus 10, 10 to the minus 12, 10 to the minus 15, and so on. And you see that this histogram is not uniform, and it varies over 20 orders of magnitude. So you have some cells that are quite easy to generate. Remember, you're generating of the order of 10 to the 9th, and some that are really hard. Okay, so these are ones that so the other thing that we've shown, and I don't have a graph to explicitly show that, is that this process is incredibly universal in all of us. So we've now verified this on over 1,000 people, and I know that each one of us has the same probability of generating any of these T cells. So the T cells here are ones likely to share, and the T cells here are the ones that are unique. And it's these ones here that help identify the murderer. So in fact, the model is so precise that we can predict exactly how many T cells, unique T cells, each one, any pair of us, or even how many T cells will share between 1,000 people. So this is the number of people. So we can ask how many T cells are shared between 2 people, 10 people, of the order of 1,000 people, with the data in blue, and the model in red. So the model is just asking what's the probability that these 1,000 people only produce these T cell, and then that it passed selection. I won't tell you in details about the selection model, but we also have a similar kind of selection model that we build on the in-frame data. And so you may ask, as humanity, with there being of the order of 10 billion of us now on the planet, have we exhausted the diversity of the system? Have we produced every T cell possible? And the answer is no. So this is the prediction of the model, and how many different T cells, basically each new person adds to the pool of existing T cells. And you see that it's not plateauing. That means that we're still generating novelty at the same rate, This is the blue is what we've actually sampled experimentally, and this is extrapolation of the sampling. We don't actually suck out all the blood from people when we do these experiments, so that's why there's a difference. But you see this system has an essentially infinite ability to generate diversity, and we're nowhere near exploiting it. And it's thanks to that, again, that an identifier matters. Okay, so since we did the sequencing on these people, we also identified that Pat and Terry had COVID. And so imagine this was like 2 years ago, and people said, well, you know, when you get a COVID test, you're actually sampling antibodies. So how can you say anything from B cells? Antibodies are B cells, but it turns out you can. Of course, there's a signature also in T cells. So in this experiment, this is actually 2 people who did go on a skiing holiday in February of 2020, came back with COVID, and then we got the data at days 15 to 85 after infection, sorted out the T cells and sampled them. And miraculously, we also had data for these people a year and 2 years before infection. Additionally, I'll add that they underwent mild symptoms. They were not hospitalized, nothing serious happened. So we wanted to see whether we can identify T cells that are responsive to COVID. So this is what the data looks like. This is the abundance of different clone types as a function of day. And you see it's a mess. What you'd like to see is a peak somewhere here, but it's actually very noisy, very messy data. So the first thing we did a PCA analysis on the time traces, and we were able to identify 3 clusters in both individuals. The purple cluster are non-responsive T cells where nothing happened. We got the green cluster with a peak at day 15 post-infection, which is what we were expecting. We also got this peak at day 37 post-infection, which other people have seen since, but we don't really understand. There's a secondary peak. We were mainly worried about how much we can trust these results, because the data is very noisy. So if you see here, we have these 2 X's also. That means we have same day, same person replicates. So if you look at 2 samples from Terry at day 15 and look at the abundances in each of these samples, you see it's very noisy. This is supposed to be a straight diagonal line, and you have some cells that appear once in one sample and 100 times in the other sample. So what we realize is that we need to calibrate a noise model. And the noise model is not simply due to experimental noise or sampling, because again, as I said, we sample the repertoire, but it's also due to the fact that there is inherent biological noise. This experiment is done on as part of cDNA sequencing, so it's based on mRNA. Sampling and mRNAs are expressed in bursts. That means that in one cell, although you have a receptor, you can have much more mRNA than in another cell. So we take into account these 2 sorts of models, sampling as a Poisson and mRNA expression as a negative binomial. And we basically go from the underlying frequencies of different cells, which we know is a power law, to what we actually observed in reads. And this noise model, so we learned the noise model based on same-day replicates, and this noise model is what allows us to then probabilistically infer a response, basically say what is a response. So a response is actually a cell that goes from an initial frequency of F2 to a later frequency, so basically expands. We assume that some fraction of cells expand and some fraction of cells don't expand, and then we use the a posteriori probability to identify responding cells. And it works. We identify a large expansion. We identify this contracting green trend we had before with a peak at day 15. Now I'm adding the -1 and -2 year data points to show you that it actually is. It's not simply a clone that went down, but it is actually an expansion and then a contraction. And we also get this secondary peak at day 37. So what can we learn from this? First of all, if we look at the memory cells, so after T cells expand, after an infection, some fraction of them is kept aside in a separate homeostatic memory pool. So we see that there is stable post-infection memory, but we also see, more interestingly, that these memory cells that are responding already a year and 2 years before the infection. So these people had pre-existing immunity. And if we look in more detail at these memory cells, we see that this post-infection long-lived stem cell memory-like cells, so these are the pink ones. So that means this long-lived memory, which was something that was and continues being disputed in T cells. Most post-infection cells are effector memory, means they're active. However, most pre-infection cells are central memory, meaning they're not very active, and then these same cells go into effector memory, so these same sequences, that's how we know, and become effector. So they basically get activated. But you could ask, so far I've showed you these are cells that somehow get activated, somehow responding, but how do we know they're responding to COVID? And this is a fair question. So for that, we did a Tetramer study. A Tetramer study is basically a molecular fishing experiment. Well, we didn't do it, but it so happened that Pat took part in day 20 after the infection in a Tetramer study. So the same person, one of our subjects. And so basically you put in a molecular fishing rod and you pull out the T cells that are specific to one specific epitope. So here they put one specific COVID protein and they pulled out the T cell in PAD that at day 20 responded. And then we could trace these T cells in our data set at different days. And so we again recovered the same trend. And in green, so we get this contracting trend. Remember, this is just one epitope, so we're not getting all of our T cells. But we can ask whether the T cells they see, we see. And in fact, all the green ones are the ones that we identified as responding or by our computational method. So we're getting all the high frequency T cells responding simply from our computational method. And we know that these are certainly responsive to COVID. So then we took this a little bit further and we looked at another study, the mirror study from Adaptive, where they did basically the same kind of, well, a similar kind of a high throughput fishing experiment where they put many SARS-CoV-2 proteins and tested them against many samples of blood. So now they pull out the T cells and they know which that are responsive to specific epitopes and they know the epitope. So we mixed their data with our data and we built similarity networks. And so we basically built networks of T cells that differ by at most 1 or 2 amino acids. And so now we have clusters and we know what clusters, what epitopes these clusters, what SARS-CoV-2 epitopes these clusters correspond to. And I won't tell you about everything, but let me focus on the big one here, which is PAD's immunodominant epitope, meaning 21% of PAD's T cell response specifically to this SARS-CoV-2 protein. And so this is the corresponding T cell that logo that recognizes it. But interestingly, these T cells that respond to this epitope were already there in PAD's repertoire a year before infection. So remember, PAD had pre-existing immunity. Maybe this is why. And a 1-point mutation in this SARS-CoV epitope gives us back a common cold coronavirus. So maybe PAD got the cold, and maybe thanks to that, then Pat got the mild SARS-CoV-2 infection. This is what we'd like to figure out in more detail now. So the last thing I want to say is basically just by looking at people's repertoires, can we figure out whether they had COVID? So for that, we used 2 publicly available data sets, one from people who had COVID and one from people who didn't have COVID. And we focused on one of Terry's epitopes. And we asked just by looking whether people have this epitope or not, can we tell whether they had COVID, that would be a true positive, or whether they, versus what's the false positive, people have the epitope but don't have COVID. And the answer is not really. But remember, these are T cells, and T cells present their epitopes on HLAs, on a scaffold that's called an HLA. And I won't tell you about this, but we didn't have HLA information, but we were able actually to infer it. And once we restrict these people from these data sets to the same HLA that Terry had, we're actually able to say who had COVID and who didn't just by looking at the T-cell repertoires. It's an expensive way, so I don't recommend it as the thing to use in your local pharmacy, but it works. So if we go back to the fact that we can predict how many sequences anybody shares, we can do it for T cells, we can do it for B cells. But if we look at people who recently had COVID, they share more B cells than predicted by CHAPS. So there is canalization of the B cell response that is visible in the repertoires. So that's it. I tried to show you how studying repertoires, we can actually make predictable predictions. Probably the most important message of my talk is don't kill anyone, even if you have an identical twin. And this may seem like an abstract problem, but apparently it is a big problem in forensics, twins that do evil stuff. So we're working with people who are seriously interested in that now. And I showed you that we can have pre-existing immunity against COVID. So thank you.
Thank you so much, Aleksandra, for a beautiful talk. I think we had two questions that got their answers in your last few slides here, but I can follow up on one of them at least. I think you mentioned briefly on one of your slides that this will also work even if you have very extended time periods. I would assume a blood sample left in a year earlier or so. Is there any limit to this, or would you expect that this is going to work no matter how old the samples are?
So we've computationally showed that it should work on the timescale of about 30 years. So basically, this is a question on how fast is the turnover of the immune repertoire. And we're finishing up a manuscript on that. The turnover of the T-cell repertoire depends on age. So as you get older, it starts turning over more slowly. But if you're in your 20s, you actually have a pretty fast turnover. So there is a limit to that, but even if the murderer is 20-something, 20 years later, they could actually be identified because the turnover is not that fast. The question though, and we've also shown on identical twins. So identical twins that share a placenta, they actually share a bloodstream in utero. And so they really just have they get given a subsampling of T cells. And we can show the signatures of that well into the 30s and 40s. So these in utero clones live for about 40 years. So these are the same sort of arguments that this is very stable. The forensic application is somewhat limited by the fact that you still need to extract mRNA from the blood. And so if you haven't done it in the right way, that's the main difficulty. And that's mainly what we're working on trying to overcome right now. But at the level of the repertoire, it really is very simple.
But relate to these twins so that even they probably won't have committed any murders in the utero. But I guess this is also going to be related to how they actually develop and adapt to their environment the first year of living, right? So can you somehow track how quickly this profile, because when they are born, they are actually identical, and then they start to separate as they experience different environments.
So that's exactly what happens, exactly what we've shown. So we have these curves. So, they were not sampled. Obviously we don't have data from people that are 20-something now from 20 years ago, but by looking at the clonotypes that they have now, so how did we do this? So it turns out that in utero, the enzyme that does these random insertions is severely downregulated. So the clones that you produce in utero have less insertions, so are very identifiable. So we were able to identify them, trace their dynamics, compare their dynamics now, and so based on that, we were able to say something about the dynamics. So so when they're born, they do have the same repertoire. More or less. and then it diversifies, and it diversifies very quickly. I mean, basically, the way you should think about this is we all have the same machinery, but we all generate different random realizations with the same machinery, right? Twin or no twin, really, we have the same machinery. It's just in a way the twins start off with generating roughly, with being given some sort of small subset that is the same, but very quickly they diversify. And since we don't actually, so there's also a question of sort of filling up, there's a question of homeostasis, and we don't fill up a T cell repertoire in utero. So in that sense, twins are not very different from us.
Thank you. We had one more question here from Rodrigo Cervera, whether you've tried the same approach for BCR since they do not rely on HLA, so it has one less variable to take into account.
Yeah, you mean for identifying, is for seeing whether people had COVID or not? Yes, and it also works.
Cool. With that,
I mean, as you could see,
yes.
sorry, may I can just add, as you could see
sure.
in the last plot, people who had COVID will share many more BCRs than people who didn't have COVID. So it's actually quite easy.
Yeah. Thank you so much, Aleksandra. I think the entire audience joins me in thanking you two for a wonderful talk. And that brings us to our last speaker for this session, but certainly not the least one. And it's an old colleague of mine, Arne Elofsson, who has been one of the pioneers in protein structure prediction and understanding quality of protein structure prediction. And as so many others, he is working on machine learning methods to do this now in particular interactions. Arne, it's great to have you here, and we look forward to hear more of your research.
Hey, thank you very much. And yeah, I'm glad to share what we have done. And I mean, for all of us in the protein structure prediction field, the last year has been kind of overwhelming, as already Ewan told. AlphaFold kind of shocked us all a year ago, and then we were very, very pleased with the release of the code, the usability of the code that DeepMind provided. It was really changed many things, and I think they also hopefully, we also provided some feedback to them, and they could improve things by that, and also certainly the collaboration between DeepMind and EBI to enable the general usability of AlphaFold has been, I think it might have been a big show of like how open source, open data is useful for many parts of science. So I will tell you a bit about the background and a bit about particularly what we have done and what we are doing right now. And so the last part is a paper submitted 2 days ago. So it's not, most people have probably not heard it yet. And it's very much done by people, both Patrick and Gabriele, but also other people in the group. But so I should not acknowledge myself very much in this. So we have been interested in protein interaction for some time. We started even before, and we realized that all the progress made in protein structure prediction can be used for interactions. So let's start. Yeah. So basically, let's start with what AlphaFold 2 or AlphaFold, as it's called, did. So this is like the results from CASP, so this protein structure prediction context, and for the hard targets, for the difficult targets. And basically, you see like the last 10 years, it was some progress. It was actually clearly progress made, but it's not so easy to see from the results. But then in 2018, you saw a jump, and then in 2020, you saw a big jump again. So basically, in 2020, the results for the hard targets were as good as for each target, and you're getting close to the accuracy of prediction of single domain proteins, which is similar to experimental accuracies. So you are really starting, consider is it just a crystal artifact, or is it really, or is it flexible loops? You're there, at least for single domains. Multi-domains, bit worse, and big complexes was not shown in CASP 14. And you can also see that other groups also caught up. So basically, but they were basically 2 years behind. So basically, the best groups in CASP 14 in 2020 were as good as AlphaFold was in CASP 13 2 years before, and then it jumped up. So it's been like, it's not one shot, it's really progress for the whole community. And one of the key things here is like, it makes interest from a machine learning point of view, is the what they call the Evo former. So that's the method to take a lot of tricks from machine learning, applied very specifically for a protein structure prediction context, and then, spending time optimizing it. So one thing you can see here is that what they do, with all of these methods, they start on the idea of you have a multiple sequence alignment. So using some method to get multiple sequence alignment. And then you look for co-evolution patterns and other patterns also, but a very, very large fraction of the co-evolution patterns. So the idea is an old one from the 90s, that if two residues in the protein are close to each other, they tend to co-evolve. So if you have a big residue interact with a small one, and this big one mutate to a small one might mutate to a big one, so you fill out the space. But then of course it's a complex network, so A might interact with B and B with C, and you see an interaction with A with C and so on, so you see all network all together. And there were methods developed maybe 10 years ago by my number group, and Martin White is one of the big developers. They call DCA methods that use that and clearly improve these methods. But in the last 6-7 years, it's been really machine learning methods can use this in the best way. So what DeepMind did was that they took this MSA and they actually threw away all this earlier work and used a method developed partly in Linköping by my ex-student, and using the raw multiple sequence alignment. So they are using basically they let machine figure it out all it. And then they used a very important trick from machine learning later. It's called attention. So basically, it tells the machine learning algorithm to focus on some particular parts. So they have attentions here, both on the columns and on the rows. So the columns means what two residues are important. So are these in context? Are they important for the prediction of something? But also the rows, because some sequences provide more information than other information. And they do this, and they have two parallel tracks. They do this, and they jump between these two representation, the representation of the MSA, and sort of a contact matrix, which is some high dimensional space. And they do this 48 times. And then at the end, they also have a structure model, so they take this pair representation of the MSAs and let the machine learning build a model. But actually, if you just skip the last part, you're quite close to performance. You're far ahead of anyone else, but it helps a little bit. And this results in models that, I mean, even this plot here up to the left is probably the most impressive. This is the RMSD, so it's the error of each atom, or the alpha atom, the backbone residue in the test. And they have like an average of 1 angstrom. All other methods have 3 angstrom. So it's really, you have details. You can get details about binding to an ion, a zinc ion here. So you see the sizes are correct, although the zinc is not in the model. So the machine has learned that this type of residues should form something that could bind a zinc. So we form the residues around something that should fit the zinc, although the zinc is not there. And then also the method works for quite big proteins and so on. So it is quite useful and it's been published and shown that it works very well. Yes, this is the key of this EVA former. And what the model, also some novelty, but that's it's more maybe of an interest for the machine learning community. It's really a new way of representing protein structure. They don't bother about anything but bond length. All of these things are learned in the model. So actually, it's almost like a gas phase that comes together. But of course, it's easy for a machine to learn that two residues next to each other should be next to each other. But this also enables us to play around with this, because we can, for instance, use 2 chains of a protein and do the same thing. We just put in a gap in the sequence. So this was published last summer, and the community went on to look at different utilities in this, and it sort of come out to some consensus what the limits are and what we can't do. So one thing is clear: we cannot really use this in general to predict if a protein folds or not. So it's not if you make a mutation that makes a protein not fold, you will get the same structure anyway. So you will not use it like that. There are probably some tricks you can start using nowadays, and there are some ideas how you can do it, but in general, you cannot do that. But of course, you can use the model and then predict the energy with some other program like FoldX or something like that, and get the idea of this. There are in general, not a very good way if you want to study dynamics. If a protein has two structural shapes, you can't really, in most cases, you only get one of them. And which one you get can be a bit random, but it's probably one that is most prevalent in the structural database. There are also some tricks that people play around to try to get to these two states, but it's clearly not obvious that you always get all the states. Clearly, a case is that when you have very large or flexible or intrinsically disordered proteins, the predictions are bad. So like this up in the right corner is nebuline, which is a very, very long protein which has small repeated helical units. It's certainly, this is from a database, it certainly does not look like that. It's actually a long, long stretch protein that's interacting with muscle fibers. So it should not look like a big glob, but this is the model you get out of it. And the coloring here indicates that the model is bad because it's yellow and red, but it's not super bad. But the overall information about the arrangement of each module is probably bad. There are some problems also because this method does not take into account other properties. So that if you have a big RNA molecule there or a big membrane in the process, it doesn't really take into account. So you get, like, particularly if you have a single spanning membrane protein, like in the bottom right corner, you have this helix location in relation to everything else is not correct. And yeah, so there's some limits, but it's certainly, so you have to treat the results with a bit of carefulness. But it's a fantastic tool for people. So the good things about it that people use it for many things is, like, of course, people use it to, one of the most valuable things is people use it for solving experimental structures. I don't know how many structures were solved that people have been fighting over for years by taking a model, doing molecular replacement in X-ray or refining a cryo-EM map. It had lots. There was a fantastic paper by Jon Kosinski about the nuclear pore complex, where they used both Rosetta fold, which is a similar tool, and alphafold to model the dynamics of the nuclear pore complex, which has like 1,000 proteins in it, which it couldn't do without it. And of course, it also predicts things like it can be used for anything you use a structure for normal. So like anything you do, like if you want to predict the effect of a variant, you can use this model if it's good, and do basic, if the model is good, it's as good as the crystal structure. So you can predict the effect of a variant. You can get some new insight in the structural space, what protein structures exist, what proteins do not exist, how common are they, etc. And of course, the models are often, at least in some cases, good enough so you actually can use it for drug design. And yeah, so it's a lot of usefulness for it. And okay, so we want to study using it for studying protein-protein interactions. So often you think of protein-protein interactions as sort of like interaction networks. You have hub proteins that interact with many other proteins, and some of these, maybe the red ones here, you know the structure of, but you don't know the structure interactions. Some you don't know the structure of. So this is a big lack of knowledge that we have. I mean, we're lacking a lot of knowledge, and we ask the questions: can you use AlphaFold to provide new insights here? So we know that we start from the multiple sequence alignment, and we basically want to go to predict some interactions here. So we had done this before. We started a few years ago doing this with the older generation methods that just predict contacts or distances. So the key problem here is that if you want to predict if two proteins interact, you have two multiple sequence alignments, and we want to extract this coevolutionary information. The problem is that we need to know exactly what two proteins interact. It's not like they just merge them together, because we want to know that that protein from that species interact with that protein from that species. If you only have one copy of each protein in each genome, it's easy because you assume that they all interact the same. But in most particular eukaryotic proteins, you have many, many copies of them. So you don't know if they all interact with each other, or if there's specific pairs that interact, or if it's one protein that interacts with all the others. But so really the interaction patterns differ, and that's if you get noise there, these weak signals you want to get out of it are often lost. So we did this. So like this is like this is pre-AlphaFold. So it's using a program called TR Rosetta and predicts the distances with proteins. So this is two proteins, like one big one up in the left corner and one small one in the right corner. And the top right part is the real interaction distances, so the contacts in the real protein, and the bottom part is one that we predict. So you can see that in both these cases, the predictions are very good for each protein by themselves, but the contacts in the interface are completely wrong. So you see the result here is that we get each protein is correctly wrong, but it's put in the wrong place. So the prediction is pretty bad. So we maybe get each individual protein right, but get it red. But in some cases it works. So we developed a method, and we were actually pretty. We thought it was we had a method here, and we developed a method that at least in some cases worked, and particularly if you run it several times and we get the same answer every time, we were pretty sure that we were correct. So this method, we call it the fold and dock method, because we fold the protein at the same time as we dock it. And we were pretty good at knowing when we were correct. And so this was published last year. And it was at least comparable to anything else. And all other methods use the structure. So this is really just directly from the sequence. It was comparable to earlier methods. But still, we were successful maybe in 10-20% of the cases, but we couldn't do it in large scale. So at least there were some examples here. You see, we have a good model. I mean, it's the below one to the right. One of the examples is the crystal structure. One of the examples here are the predictions. So at least in some cases, it seems to work. Okay, so now we want to do the same thing when AlphaFold was released. We want to do the same thing. So basically, we take two alignments. We can combine them some ways, and you can combine them in different ways. And we just put that into AlphaFold. And so we we did this. And of course, we were lucky to have the access to Berzelius. So we had this big computer, so we could do it quite fast. So we managed to do it before most others. We made a benchmark, and particularly played around with how to get this alignment the best possible way out. So we did that, and you can see, like, if you use these other methods, we have like, this is a plot of the dock Q score, which is like the quality of the docking. And you can see with all methods, we have basically nothing correct. Off like 0.2, 0.3 is the correct benchmark. And if you run AlphaFold 2 default, we did better. We had some good models, but still the average was pretty bad. But if you start playing around with alignments, we could push it up to like 60% of models were higher than our cutoff. We really could, by playing around with the alignments, we could improve the prediction quite a lot. So what we did was that we basically, you would expect that as we did before, that you take the alignment and you try to merge them together. We call it pairing. That would be the best way. But actually, it was shown that if we also included what we call the blocked alignment or the fused alignments, that where you basically just take one sequence and put it to a larger gap on the other side, and we put them together, then actually that is the best way to get results. And you can also run the fused sequences, because there are many cases in proteins where you have actually a fusion between two sequences. So if you just put the sequences next to each other, in some cases you get a hit that covers both sequences. So if you just combine all this together and then do some ranking at the end, you can actually get up to 63% of the models that are tested that were correct. Then, of course, DeepMind realized also this was a good idea, so they released a method called AlphaFold Multimer that does a similar thing, but it does it all internally to AlphaFold, and that pushes the results up to like 72%. But that is still in a preprint form, because they had some problems I'll come back to. What is more important, I think, was that we actually, not only did we do good predictions, it's actually we could tell when we were correct. We just took a very, very simple formula. We took the quality of the residues in the interface and multiplied it with the size of the interface, basically number of contacts. And we did that, and we found that it has like a sigmoidal fit to the quality of the docking results. We could get a good value between 0 and 1, that basically if you have a high value close to 1, or like 0.5, 0.7, you are almost always correct. And if it's around 0, you're almost always wrong. So we are very good at predicting when a model is correct, when it's wrong. And we could say we have an area on the curve, or selecting good bad models like a 0.88. And we can even use it to predicting if two proteins interact or not at all from some other data set, and then it's like it's not as good, but it's like 0.82. So we can really start working on large-scale predictions of interaction networks. So this is just some examples. So you see on the top is some nice cases where it works well. These are quite big proteins that were not in the training set, that are novels. They were not, no way that AlphaFold can learn them, and they are basically correct. But there are also some cases here. But one case we have in our DNA molecule or RNA molecule that is making the prediction being wrong, and the one that we just do a wrong prediction of. So there are cases where it works and cases where it doesn't work. So then we want to take this a step further. So of course, we want to not study pairs of interactions. We want to study the whole set of large-scale interactions. So we took some, we can't computationally run all proteins against each other, all pairs against each other. That's too computationally costly. So we had to limit ourselves to some predefined set of interactions. So we took two data sets. One is URI, which is basically yeast-2 hybrid study, and one is large scale. So there are like 64,000 interactions in that one, plus additional some literature benchmarks. And the other one is UMAP 2.0, which is a co-purification study, basically, where they basically co-purified proteins, and then you do some clustering, and then they do a picture of this. And then we took this and compared these predicted docu values that we have for all these interactions. These are like 65,000 in one case and 15,000 in another case. And you can see that if you take a random set first, we take a blue curve here, basically all those have a predicted docu value of very close to 0, a few exceptions. So it's just random pairs. If you take this yeast-2 hybrid data, you see that most of them also have predicted value of close to 0, but there's a small peak up here. If you take the UMAP data, black curve here, you have a bigger peak here with good predictions, but it still is like majority. We do not predict to interact. But if you compare that to data where we have small scale yeast-2 hybrid studies, or we have this 3D structure known of it, we have also two peaks, but most of them are in the highly predictably high, interact strongly. So clearly there is some kind of difference between the experimentally verified large-scale docking study or interaction studies with what you see in PDB or within small-scale studies. Because in these ones we have like in small-scale studies we have like 60% of interactions we can predict. In the other ones it's maybe a few percent or 10% maximum. But anyway, we can see this, and also you can see that the overlap between UMAP and URI is very, very small. It's basically random, not really, but it's close to random. So there is probably different types of interactions. And it's only one type of interactions that we are good at predicting with high accuracy. And that's probably the interactions that are present in PDB, which is like large complexes, it's like strong, stable interactions, and not transient interactions. So we clearly are missing ability to predict transient interactions. But we have some nice examples, and we can basically increase the structural knowledge of UMAP quite significantly, at least like 3,000 bigger. So we have 3,000 or 4,000 new complexes that we are pretty sure are correct. Some of these we have verified by cross-linking or by other data. So we have pretty sure they're correct. And also you can see that some are bad, pretty bad, some are good. And there probably are some good predictions that do not agree with that are novel also. But what we found most interesting was when we started looking at the network. So you have this network, and you have like blue and red dots. So the red dots are the one we can predict that are known structure, the blue ones are not. And we see that these clusters here, and some of these we have good scores for. So like in some cases, we have like very clusters that look like very much everything is interacting with everything. And some we have like a node that is interacting with many other combinations there. And this was usually called party hubs, because you're in a hub here, you have party, you interact with this is party hub here, we have interact with everybody all the time, or you have a date hub, you go on a date, but you go on a date with very, very different people. And if you look at the structure of these, this is a proteasome, the big cluster here. So basically, this is, of course, is a big complex. Everybody's interacting with everybody else, or many interact with each other all the time. While this one is GDI2, which is basically one GDI1 is interacting with many different proteins, but not at the same time, because all the models look the same. So basically, you can superimpose them on top of each other, so they can't interact at the same time purely physically, because they are interact the same way. So really the structure can provide you insights into why the networks has the structure it does to. Because here in the GDI1, it is impossible for two proteins to interact at the same time with the green protein. But in this case, you have part of a big complex, it's all big complex. And both these predictions provide some biological insights. So next we went on to look at the proteasome. So the proteasome, we tried to basically have models of all interactions. These are red ones, are good predictions. There are few are missing. So we thought we should, can we build a model of the whole proteasome? And then Fredrick realized that you can represent this as a network. So you can take a network, basically what interaction was, and if you just add nodes by the network, you add them one by one, you can build up a big, nice model of it. However, it's important in what order you build it up, because you have introduced small errors or things are wrong. So if you start in the wrong order, you get a bad model. But if you start in the right order and you select the right order, you can build up a very nice model that is 90+% accurate. So we tried to do that using, and first we realized it was computationally very, very expensive. So Fredrick also realized you can use the Monte Carlo tree search algorithm that is invented a long time ago, that basically gives you probability that one node here where you start from should be part of the optimal node to find at the end. So you can do that, and that means you can limit the number of possible search patterns to something reasonable, and you can then, from these small complexes, start bringing up bigger complexes. So one thing we also realized, I think you can see it here, is that if you started from dimers, it did not work very well, because you start to end up things that end up in the wrong confirmation up here, because they have basically or they start clashing with each other and so on, or you really could not build it from dimers. You only had one type of interface. You're missing one type of interface. But if you start from trimers, then you treat these trimers as 3 dimers, you have many more possibilities. So you can actually start doing here, you can start with a trimer, you can start building it around the chain here. So Fredrick ran for a set of 175 proteins, we run trimers, and we deal with 2 different ways. We have we generate all the trimers and the dimers, and you can see that if you have dimers, it succeeds in some cases. If we have trimers and we know the interaction partners, we know which proteins is in one, so it limits the number of possible graphs. That is, we get some better results. And actually, if you take everything, which is computationally much more heavier, we get about half the models are quite good, and like a small set is really, really good. And we can have a scoring function for this. We can say know when we are correct. So we basically have, if you have a cutoff or a scoring function, we can get something which is like, with some accuracy, you can predict if the model is correct or not, using the same type of score we had before. So this is basically what we end up with. So this is cases we start with trimers for 23 of these 175 complexes. We have a TM score of higher than 0.9. So basically, all the chains are built, and there are 90% of them are within 3 angstroms errors like that. So you can miss some loops here that are wrong and so on. And notable here, this is not built from the sequence of the PDB files. This is known from the sequence of UniProt. So we have residues that are extra here that are disordered, that you see end up here, but we had to put them there anyway because they're not part of the crystal structure or not seen in the crystal structure. So this is just some nice examples where we can build things. And we did some analysis on what works. Of course, it works better in bacteria than in eukaryotes, as always. And if you have more sequences, it also works a bit better. So this is done here. It seems to work slightly better for homomers than for heteromers, but it's not so strong. And of course, an important factor is if you need to predict these trimers correctly, then it works better also. Okay, so that's basically first. I want to thank Patrick and Gabrielle and Julie and Aditi and Petras for helping with all of these projects. And this network project is done together with Pedro. And then of course we are very actually grateful for this thing up in the top right, which is the Berzelii supercomputer, and to SNCC and Scilab Lab and Wallenberg and VR to provide funding for this. So Thank
you so much, Arne, for a very exciting talk. We have 2 questions in the chat here. Sam, or at least his Mac, has asked whether you could clarify a bit how the MSA is encoded for AlphaFold. Presumably the number of channels need to be fixed somehow.
So they are, no, it doesn't have to be fixed. I think they have, I think they do, well, it probably, if I remember correctly, it's fixed, but they do subsampling or randomness of it, I think sometimes, if I remember correctly. So I don't think they might have some fixing there. I can't really remember, to be honest. I have to look in the paper.
Marta Caroni also has a question. How much more difficult do you envision it to be when you have to predict interactions between homologs that make up a functional complex, such as, for instance, the DNA helicase?
So one of the limits we have really, like, which is actually, you can look at the proteasome here, which is similar, because that basically the proteasome are 14 unique proteins in the bottom part of it, 2 copies of each, and of course they are all homologs. They basically all have the same structure. So and at the moment we have no good way of separating which of these proteins interact with each other. So if you make a model of these 14 proteins, well, it's 28 proteins in 2014, we will put them in the wrong order because they are too similar to each other. So to really selecting out which one is interacting with which one, you will need to do something else than using AlphaFold. You'll probably really look at the interfaces and calculate the energies or something like that. That's a limit at the moment. And we tried some simple ways of scoring it, and it doesn't seem to correlate very well. So it's not. So that's clearly its limit.
And finally, we have a question from Ola Larsson, whether your methods can be used to exclude interactions.
I would say that it's. I mean, if you look at our predictions of here, this is basically a data set of non-interacting and interacting proteins. So certainly, I think we are. I mean, it's pretty good at separating interactive non-interactive proteins. Of course, if you take all pairs of proteins, that's a large number. So you wouldn't say they're completely excluded. And I think we can do it to if we include only discuss the type of interactions that are really stable. So I think like if you really take that these 2 parts are not interacting strongly with each other in a complex, yes, that's probably doable to some extent. If they have a transient interaction occurring sometimes in cell, then we can't do it. So it really depends on the interaction type.
Thank you so much, Arne. And I think with that, we are exactly at 10:55. Thank you first to Arne for giving another exciting talk. Let's thank him first. And then let me thank all the speakers for staying perfectly on time, because that means that you all now get an opportunity for a 10-minute break before we're going to reconvene and have Janne Lehtiö take you through the session for precision medicine. And I'm going to be back and talk to you later this afternoon. So see you in 10 minutes.